An important part of this section is understanding how to answer questions using amino acid tables. So you would be given these. You do not have to ever learn them. You don't have to learn a single name of any amino acid. You simply need to learn how to use these tables. So if you look, you'll get a couple of different forms of tables. The first very important step is, what table am I given? Am I given a table with DNA triplets, mRNA codons, or tRNA anticodons? So this will always be said in the instructions or at the top of the table, for example. So in this case, if you're given this table, the instructions would say use the table of DNA-based triplets below to answer what amino acids are in the molecule or whatever. So in this case, we definitely know it's DNA because there's thymine. As soon as you see thymine, there's thymine as well. That is going to be a DNA type question. So all you do is you see here, this is the first base. Now this is a more complex table. We'll look at some simpler ones. This is the second base. So this is the first letter, that's the second letter, that's the third letter. So if you have the code, let's say TAC, I'm just going to write that over here for you. And they want to know what amino acid that codes for. T is the first letter, so it's somewhere in this row. A is the second letter, so it's somewhere here. And then you just say, oh, well, there's C, which is the third letter, TAC. So the amino acid is tyrosine. But you just write TYR. If they haven't given you the full name, you don't have to know that tyrosine is TYR. If they've given you an mRNA table over here, this is an mRNA table. Again, if the code is something like AUC, you go first letter is an A, second letter is a U, and the third letter is a C. You find AUC, and that is going to be the amino acid ILE. I'm just trying to point to it there. So AUC is ILE. And again, you don't have to know what that stands for. If you get an amino acid wheel, this is less common, but it does happen. They will give you instructions on how to read it. But all it is you read from the center going outwards. You follow the arrows going out. Okay, there's arrows. So again, if the code is AUC, you start with A, then you go to U, then you go to C, and then it's ILE. It's the same amino acid. So we get the exact same answer, just using a different method. So the next step would be to translate, in inverted commas, it's sort of related to translation, the three letters that you're given. So it might be ATA, it might be ACU, it might be AGA, whatever it is, but it's the three, it's either a base triplet, a codon, or an anticodon into the given amino acid table. So what I mean by that is, if you have a tRNA anticodon, and that tRNA anticodon is, let's just say, ACA. But you've been given an mRNA codon table. You can't just plug in A, C, A, and then match it to that amino acid there. You need to convert, because you've got an anticodon, ACA, but an mRNA codon table, you first need to convert ACA into a codon using your complementary base pairing. So A would be matched with U if we were looking at the anticodon being matched to a codon on mRNA. The C would be matched with G, and the A would be matched with U. So actually, the mRNA codon is UGU, and then you use the table you're given. So it would be U, and then you find the second letter being G, and the third letter being U, and then it's going to be cysteine. Or you have your amino acid wheel, and it's UGU, and it's cysteine again. So you must be careful to see what information you are given in the question versus in the resource of the table. Let's look at a couple of examples. So this question comes from June 2018, paper 2, matric paper. So the question is, I always read the blurb first very carefully. Insulin is one of the hormones responsible for the control of blood glucose levels in humans. Now, the first thing I notice is that hormones are all protein in nature. So that means they're going to be made through protein synthesis in the same way of transcription and translation. It consists of two long amino acid chains, A and B, which are joined. So chain A is made up of 21 amino acids, chain B is 30 amino acids. The diagram below represents the amino acids present in each chain. So this here is a diagram of the insulin protein / hormone. So you can see very clearly where is chain A and where is chain B. So I would take my highlighter and just highlight chain A and highlight chain B in a separate color. So chain B there is 30-something amino acids. Name the process whereby insulin is produced in a normal human cell. That's going to be protein synthesis because insulin is a protein. So 6.1, I'm already knowing the answer there, is protein synthesis. 6.2, identify bond X. Because this is a protein, that is a bond that is joining two amino acids. So that will be a peptide bond. We can just call it peptide. The tick will be on the word peptide. You don't have to say bond. It's redundant. How many nitrogenous bases in a DNA molecule code for the amino acids in chain A? Now, guys, these questions are difficult because we get confused. So if we just remember, if we have a DNA molecule here and 1, 2, 3, 4, 5, 6, 7, 8, 9 bases. So I'm just going to write here that's DNA. That would mean an mRNA molecule here would also have 9 bases. So that's mRNA. And I'm just drawing them in threes because that is a codon and that is a codon, etc. So all of those are codons. These would be considered base triplets. And each of the base triplets and the codons are made up of 3 nitrogenous bases and 3 nucleotides. Then you'll have tRNA. That's the wrong color. Then you'll have tRNA coming here. And there will be a molecule there with an anticodon, a molecule there with an anticodon, and a molecule there with its funny Deutsche shape. Funny Deutsche stickman shape, funny Deutsche stickman shape. So in the end, if you think about it, each of these tRNA molecules carry 1 amino acid. So I'm just going to label that there as being amino acids. So if you break it down like simply, not worrying about how many amino acids are found in insulin, there are 3 amino acids here, 3 anticodons, 3 codons, and 3 base triplets. So the ratio of amino acids to codons, anticodons, and triplets is 1:1 because 1 amino acid is carried by 1 tRNA molecule. But that means that for every 1 amino acid, there have to be 3 nitrogenous bases on tRNA, 3 nitrogenous bases on mRNA, and 3 nitrogenous bases on DNA. So that means for every amino acid, there are 3 bases on DNA, if we refer back to the question. So in this question, they're asking how many DNA bases are there in the chain A. So chain A is made up of 21 amino acids, and for every amino acid, there are 3 bases. So it's just 21 times 3. So that will just be 21 times 3 is 63. So the next question says the table below shows mRNA codons that code for some amino acids. So it's the mRNA codons being given. Name the nitrogenous base represented by G in mRNA. Now that's an easy one. It's just going to be guanine because that's what G is. The next question is a little bit harder. Reading the chains from left to right, give the codon for the 4th amino acid in chain B. So you've got chain B, the purple one, 1, 2, 3, 4. You're reading from left to right. So that is GLN. So you read here, you find GLN, and that's glutamine. And now the question here, you don't just write what glutamine is, you say you need the codon. So here you simply read it from the table, and that's going to be CAG. So you just write CAG. The next question is the DNA base triplet that codes for the first amino acid in chain A. So the first amino acid here is GLY, which is glycine. So this is how I work it out. I'm just going to do it in red so you can see it's going to be working out here for question 2. So in the table we have mRNA, and the mRNA code for glycine is GGA. So it's this one over here. Now, mRNA is made from DNA during transcription. So then I will just go work backwards and say, well, G would be bonding with C, G with C, and A will be with thymine. Because remember, DNA has thymine. If it was going to tRNA, then that A would bond with a uracil. So then your answer would just be CCT. Now, that is a sufficient way to answer the question because you're showing DNA and mRNA. You're labeling this as DNA, so therefore I will put the tick on CCT. But you can also then write, if you want, therefore CCT as your final answer. Then your final question here, give the anticodon for valine. So you've got GUA is the codon, and I'm going to do the same working out here. So you've got mRNA codon being GUA. So I'm just going to highlight it here. And then that goes to tRNA. So tRNA, if it was a G, it'll be a C on tRNA. If there's a U on RNA, it'll be an A. And if there's an A, it'll be a U. Because remember here, there was an A and it was a T on DNA. But we're on tRNA now, so it's a U. There is no thymine. So your final answer there, you can therefore write, therefore, CAU as your final answer. So it's reading questions very, very carefully.